Hi, today we're going to talk about aerobic respiration. The reactions of this process occur in various locations of the cell. The mitochondria is one of the main locations and is a very important organelle involved in aerobic respiration. Mitochondria are shaped perfectly to maximize their productivity. They are made of two membranes. The outer membrane covers the organelle and contains it like a skin. The inner membrane folds over many times and creates layered structure called cristae. The fluid contained in the mitochondria is called the matrix. The folding of the inner membrane increases the surface area inside the organelle. Since many of the chemical reactions happen on the inner membrane, the increased surface area creates more space for reactions to occur. The intermembrane space is located between the outer membrane and the cristae and contains many protons, the importance of which we will discuss later. So now we can dive into the first reaction of aerobic respiration, which is glycolysis. It actually occurs outside the mitochondria within the cytoplasm of the cell. Glycolysis is separated into two phases, known as the preparation phase, which are reactions 1 to 4, and the payoff phase, which are reactions 5 to 9. We will talk about these in more detail later. Before glucose can be moved into the mitochondria, it has to be turned into pyruvate, which occurs through a series of reactions. So let's see how this happens. Firstly, glucose turns into glucose 6-phosphate. We will call it G6P in short from now on. This process requires 1 ATP. Glucose is phosphorylated by the enzyme hexokinase to become G6P, using the inorganic phosphate from the ATP. Then G6P turns into fructose 6-phosphate. Fructose 6-phosphate is then phosphorylated by the enzyme phosphofructokinase 1 into fructose 1,6-biphosphate. This reaction requires a hydrolysis of 1 ATP. Reaction 4 is when fructose 1,6-biphosphate splits into 2 glyceraldehyde 3-phosphates, therefore doubling the production of ATP in later reactions. This marks the end of the preparation phase. The payoff phase begins with reaction 5, where glyceraldehyde 3-phosphate turns into 1,3-biphosphoglycerate. In reaction 6, 1,3-biphosphoglycerate turns into 3-phosphoglycerate, releasing an ATP. The following reaction is where 3-phosphoglycerate turns into 2-phosphoglycerate. In reaction 8, 2-phosphoglycerate turns into phosphoenol pyruvate. Reaction 9 is important as it is the final limiting step. In this reaction, phosphoenol pyruvate turns into pyruvate by the enzyme pyruvate kinase, producing 1 ATP in the process. In glycolysis, a total of 4 ATP are made, but 2 are used up in the process, therefore the net gain of ATP is 2. The next step is the link reaction, which is fairly short. It starts off with pyruvate and coenzyme A in the cytoplasm. They then combine to form acetyl coenzyme A. This releases a CO2 and a reduced NAD is formed. Acetyl coenzyme A then moves into the matrix of the mitochondria and separates into acetate and coenzyme A. Coenzyme A then moves back into the cytoplasm to be used for another reaction. So now we are going to talk about the infamous Krebs cycle, also known as the citric acid cycle. It occurs in the matrix of the mitochondria and uses the acetate formed from the link reaction. There are 8 steps in the Krebs cycle, which are split into the regeneration phase, which are reactions 1, 2, 7 and 8, and the oxidation phase, which are the remaining reactions. The main goal of the regeneration phase is to allow acetate to bind with oxaloacetate to form citrate, which happens to be the first step of the Krebs cycle. Citrate then turns into isocitrate, which is the second step of the Krebs cycle. The oxidation phase begins with the third reaction of the TCA cycle, where isocitrate turns into alpha-ketoglutarate, releasing reduced NAD and CO2. In reaction 4, alpha-ketoglutarate turns into succinyl coenzyme A, which also releases reduced NAD and CO2. In the next step, succinyl coenzyme A turns into succinate by releasing ATP. Succinate turns into fumarate, releasing reduced FAD. This marks the end of the oxidation phase. The regeneration phase begins again in step 7, where fumarate turns into L-malate and the finished L-malate converts into oxaloacetate, ready to combine with acetate to start the cycle all over again. For each glucose, this cycle occurs twice, producing 2 ATP, 4 CO2, 2 reduced FAD and 6 reduced NAD. This is the net equation. Last but not least, we have the electron transport chain, which is the final step of aerobic respiration. The purpose of this reaction is to return reduced NAD and FAD into ATP. This occurs by both reduced NAD and FAD donating an electron into the electron transport chain, becoming NAD plus and a proton, and FAD plus N a proton. As the electrons move along the electron transport chain, the protons released by the reduced NAD and FAD accumulate in the intermembrane space, creating a concentration gradient. Protons then move down the concentration gradient via ATP synthase, causing a conformational change in the protein and leading to the production of ATP. Oxygen acts as an electron acceptor at the end of the chain to create H2O, allowing the cycle to start again. At the end of aerobic respiration, the net gain of ATP is 34 as 4 ATP are used in the process. Now that you know exactly how aerobic respiration works, you can now appreciate the process your body goes through to keep you alive and kicking. And remember, mitochondria are the powerhouse of the cell.